Hello, everyone. This is Dr. Richard Fleming, and today is Saturday, the 27th of February, 2021. For those of you who've had an opportunity to watch me on my YouTube channel, you realize that following the emergency use authorization of the vaccines for both Pfizer and Moderna, I reviewed the emergency use authorization, the EUA documents, submitted by those pharmaceutical companies to the FDA. Yesterday, I, like many of you, undoubtedly watched the emergency use authorization discussion by the FDA for the Johnson & Johnson vaccine. I thought, given several of the questions that have been posed to me over the last 24 hours, that I would review the emergency use authorization for the Johnson & Johnson, or sometimes referred to as the J vaccine. One of the important distinctions between these vaccines is that the Pfizer and the Moderna vaccines provided a lipid nanoparticle coating of the messenger RNA. The Johnson & Johnson vaccine, like the AstraZeneca vaccine that's out of the UK, yet to obtain its emergency use authorization, are slightly different. These two vaccines use another virus. It's called an adenovirus or adenovirus, depending upon your pronunciation, where you're at in the world. And it incorporates inside the virus not RNA, but DNA, double-stranded DNA. This changes the conversation somewhat because in the prior discussions of the Pfizer and Moderna vaccines, questions were raised as to whether the genetic material encoded in the vaccines could get into the nucleus of our cells. And for those of you who have followed the information on FlemingMethod.com, you'll see that there's published research, or at least preprints of published research, showing that in fact the mRNA does incorporate into the nucleus of the human DNA. The Johnson & Johnson vaccine is a different matter. It is double-stranded DNA, deoxyribonucleic acid, with the expressed intent of getting into the nucleus of human DNA, from which our cells will be used to make spike protein following the insertion of the double-stranded DNA from the vaccine, with transcription, the process of transcribing the DNA to messenger RNA, and then translation, the process of taking the mRNA and making a protein, in this case, the spike protein. With that in mind, we no longer have to ask about whether this vaccine will get into human DNA or into human nucleus. It is designed to get into the nuclear area of the human cell, and from there, we will see. I want to take a moment, or several minutes in fact, to share some information with you from the emergency use authorization as I did with the Pfizer and Moderna vaccines. And together, we will go through the process of reviewing this actual document that was presented yesterday, Friday, February 26, 2021. This is, in fact, the emergency use authorization, and it's slightly longer than the Pfizer and Moderna ones, partially because it also talks about some of what's been noted with the Pfizer and Moderna. So as we scroll through here, I'm going to provide the same approach where I highlighted many areas that I considered important to review. You can see there's quite a long index, and then it gets to an executive summary. It's pointing out that there are somewhere around 50 billion viral particles in the vaccine coming out from Johnson & Johnson. Now, the specific Belgium company is Janssen Biotech Incorporated. That is a part of Johnson & Johnson, and it has the express purpose of making this vaccine. It's interesting to note that they're not suggesting that it's going to prevent you from getting infected and ending up in the hospital. Much of the discussion has to do with a confusion between SARS-CoV-2 and COVID-19. In most of this emergency use authorization document, the company that submitted to the FDA refers to the entire process as COVID-19. So it's important to distinguish. SARS coronavirus 2 is the virus. COVID-19 is the disease resulting from that virus that is both the result of the viral infection and, more importantly, as you've heard me say numerous times, the inflammothrombotic response of our immune system to this virus in individuals with comorbidities. Now, as we're seeing with the VAERS, the adverse events material showing up with Pfizer and Moderna, the inundation with billions of mRNA from the Pfizer and Moderna vaccines are producing these same types of inflammothrombotic and other responses, given the inundation of our cells with a greater amount of genetic material, mRNA, of the SARS-CoV-2 spike protein, which, as we have discussed, shows several gain of functions demonstrating that it is human-made and not naturally zoonotic. Zoonotic means naturally occurring. So if you hear that term, that's what they're referring to, zoonosis or zoonotic, naturally occurring. And the evidence suggests anything but it being naturally occurring. The information provided during phase 3 of this Janssen study looks at approximately 40,000 participants. Now, we will just discuss in a few moments the difference between phase 1, phase 2, phase 3, and even phase 4 studies, just so that you have it. I want you to know, first off, let's review this, that when they're talking about adverse events, it's graded, according to the FDA, grade 1, 2, 3, 4, and 5. So grade 1 is what's considered a mild event. That's a variety of meanings. Grade 2, moderate. Grade 3, severe. Mild, moderate, and severe. Those nice qualitative terms that give a fuzzy coating to what you're looking at. Do you consider it mild, or moderate, or severe? Excuse me. Grade 4, potentially life-threatening, and grade 5, we would all agree upon, which is the person died. So with that in mind, as they discuss some of these different grades, this is what they're referring to. It's not a quantitative or measurement scale, it's a qualitative, an estimate scale. They refer to vaccine efficacy as to whether the symptoms of COVID-19 disease actually occur or not. Now, the function of having a vaccine is to introduce an antigen, something that is foreign to your body, typically a protein, in this case, part of the virus, which will become the spike protein, to get your body's immune system to recognize that it shouldn't be there and develop an immunologic response. The first part of that response, as we've talked about before, usually occurs from days 3 to 5 in what's called a T cell response. This is the type of cells originally formed in the thymus gland, hence T cells, that are responsible for attacking and killing cells where the virus is present. So it's called the innate, naturally occurring T cell cytotoxic response. And later on, at 7 to 10 days, you have what is called a delayed adaptive humoral, which means antibody response. To determine if a vaccine is effective, if it's causing T cells and B cells, which make the antibodies, to become activated, you would need to prove that the T cells and B cells are becoming activated. You would need to show measurements of the T cells, and you would need to show what's called antibody titers to show the effect of the vaccine gearing up your immune system so that when you get the infection, naturally from person to person, you will have a shorter duration or less severe response to the infection. Nowhere in this emergency use authorization and nowhere in the discussions yesterday held by the FDA was there a discussion about measured T cell responses or antibody responses, the two quantitative measures of vaccine efficacy, and they're missing. They were missing yesterday. They're missing in this emergency use authorization. They were missing in the Pfizer vaccine emergency use authorization, and they were missing in the Moderna vaccine emergency use authorization. Those papers, just for quick review, December 10 for Pfizer, December 17 for Moderna. What they are using here is the presence of symptoms as COVID-19. Becoming symptomatic but not being hospitalized is not COVID-19. We don't speak about any other virus as being a disease. We talk about you as having the virus and having rhinorrhea, runny nose, myalgias, muscle aches and pains, becoming febrile, an elevated temperature, those types of symptoms. They don't equal disease. They haven't before, and they're trying to muddy the waters by declaring it disease now. COVID-19 is coronavirus disease, first noticed in the year 2019, that results in people being hospitalized or needing medical treatment. Prior to that, you have a virus. Now, it may be a man-made virus, but it's still a virus. They're using polymerase chain reaction. Now, we've talked about this numerous times, and there's a lot of debate and discussion about it. But the truth of the matter is that this test was designed to find genetic material. And for that, it is exquisitely designed. But it was never designed to be used to determine whether you needed treatment and or what that treatment is. So make no mistakes about it. PCR testing was approved as an emergency use authorization test. And this emergency use authorization test is now being promulgated as the defining factor for emergency use authorization vaccines. An unapproved test being used for unapproved vaccines to try to sell the concept that vaccination is the only way to address an infection, in contrast to all the evidence showing that we have treatments that are available and effective and safe for SARS-CoV-2. In general, they break this down in this emergency use authorization, looking at age, comorbidity, race, ethnicity. And one thing they noticed over and over and over again was that the people that were over 60 and had more comorbidities tended to have more problems despite being vaccinated. And the younger people tended to complain more about having symptoms related to the vaccine. And you can make your own social comment on that. What they do comment on is that the data was insufficient to assess vaccine efficiency or efficacy in patients with evidence of a prior SARS-CoV-2 infection. So when you hear people asking the questions, should I take the vaccine if I've already been infected? They begin right up front by telling you that none of these vaccines have been shown to have any beneficial effect in people who have already had SARS-CoV-2. They noted a lot of country to country variability. And in fact, much of the information comes out of the United States, Brazil, and South Africa. And South Africa, with its new variant, seems to have more challenges than other countries. And that was the same in the research study that we published, with the South African site having more challenges in following through some of the treatments and diagnostic evaluation of tissue levels. They noticed in this study a number of what's called numerical imbalances, which means there's more of a problem in the people that were getting this vaccine than people who got the placebo. Two of the major areas they noticed were thromboembolic events. That's another term for the inflammothrombotic response that I've been talking about. The thrombus, the blood clots, and the embolism, the movement of those blood clots from one area to another. So you could have a venous in a vein, thrombosis, blood clot in the leg, that when it's dislodged, goes up past the right side of the heart and goes into the lungs, forming a pulmonary embolus. So you have a thrombus embolizing, which an embolus is a moving blood clot. Or there are also something called fat embolus that occurs typically after a motor vehicle accident, when larger bones of the body are damaged and broken, and the a fat embolus forms and moves and goes throughout the body. They also notice problem with ringing in the ears, tinnitus, or tinnitus, if you wish, depending again upon where you're raised on planet Earth. The purpose of the meetings in the past with the Vaccines and Related Biological Products Advisory Committee, that is the committee looking at all this, was to determine if they could prevent COVID caused by SARS-CoV-2. They're not preventing infections, and they're not preventing you from spreading it. The goal here, every time they talk about controlling or preventing COVID-19, is to minimize the symptoms. And they're doing a good job of pushing these terms together to confuse people in a time when there is so much misinformation and confusing going on. It's somewhat disconcerting to me as a scientist, as a research scientist physician, to take this report credibly and not look at it skeptically because of this combining SARS-CoV-2 as the virus with the actual disease process, and talking about this vaccine preventing COVID-19. The goal of the vaccine is to reduce the severity of an infection, and hopefully that will reduce the severity of any sequelae or disease that one might experience. This is the confusion of the terms. This is where we comment on the fact that if you go through here, they want you to accept right up front that this is a zoonotic coronavirus. Now, I don't know if they know Peter Daszak or what connection they have with EcoHealth or the University of North Carolina or Wuhan Virology Institute. But no matter how you look at SARS-CoV-2, whether you look at the HIV pseudovirus glycoprotein 120 insert that Shang Li and Barrick admit to inserting into this spike protein, or you look at the PRRA insert, the 4 amino acids that separates this coronavirus from any other coronavirus on the planet, but is found in things like rabies virus and cobra toxin and HIV glycoprotein. If you look at the sequential change in the spike protein that occurs as a result of these two inserts, and you realize that now the regional binding domain has prion-like regions or prion-like domains in the receptor itself, you can appreciate the fact that this is not a naturally occurring virus. And the millions, multi-millions of dollars that have been invested from NIH and NIAID and the Department of Defense and FDA and Health and Human Services and other federal agencies that bypassed the restrictions and went off to Peter Daszak at EcoHealth proved worth their money as far as developing a gain-of-function virus with increased infectivity that we're seeing around the world during what is now the endemic. of 2021. It's no longer the pandemic of 2020. We have the endemic of 2021 with treatments available if physicians are merely allowed to do their job that we're trained to do. This, as I mentioned earlier, is an adenovirus that has been replicated inside human embryonic cells that were retinal cells that were taken out of an 18-week-old fetus. The virus is grown there. It's called adenovirus type 26. We are expected to take their word for it that this is a viral vector that can no longer replicate. So we have a virus that is, by virtue of the type of virus it is, this adenovirus normally wants to infect human cells. And inside that, Johnson & Johnson has inserted double-stranded DNA from the actual SARS-CoV-2 virus itself, with the express goal of putting all 50 billion viral particles into the human nucleus so that that DNA can be turned into RNA, can be turned into spike protein. They're focusing here primarily on what's called the phase 3 studies. These are the ones determining whether the treatment of this vaccine is better than anything that's out there already. They believe, by looking at the number of COVID-19 cases that occurred after people got vaccinated, that there is a 66.9% efficacy in reducing moderate to severe COVID-19 at 14 days and 66.1% at 28 days. So the statement is that it reduces the number of individuals who progress to develop COVID-19. And the ones who go ahead to develop COVID-19 have inflammothrombotic blood clots, tinnitus, and a variety of other problems that are associated with the virus itself. That's responsible for killing so many hundreds of thousands and millions around the world because they aren't being treated for the inflammothrombotic response. Now, these are, as we've looked at before, the 4 requirements for an emergency use authorization to be granted, either for a vaccine, a drug, or a test. So the first step required that the Secretary of Health and Human Services declare we have a problem that needs to be addressed. And that was done back on March 27 of 2020. The second is that what we're looking at is something that can be used to prevent, diagnose, or treat, or to mitigate a product already being used to prevent, diagnose, or treat, in this instance, SARS-CoV-2, that the benefits outweigh the potential risks, and critical, that there is no adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. Now, we have already demonstrated with the paper that we published on the 4th of February this year, outpatient successful treatment of 100% using primaquine and clindamycin and hydroxychloroquine, 97.9% efficacy with hydroxychloroquine and clindamycin, and slightly reduced efficacies, but still greater than what is being touted with this vaccine with hydroxychloroquine, azithromycin, hydroxychloroquine, and doxycycline, except this actually provides treatment while the vaccines do not. This is all actually dirt cheap, while the vaccines are not. You may get a free vaccine, but the United States taxpayers have paid millions of dollars, and I think in one instance I saw billions of dollars for these vaccines. So just because you're not paying for it when you get the vaccine doesn't mean it was free. We've all been paying for it as taxpayers. And by the way, treatment responses of those that were critically ill that ended up hospitalized were 99.83% successful in the study that we published. And I would point out also that many other physicians who have been looking at the treatment of aminoquinolines like hydroxychloroquine and the antibiotics, although that's not how they're working for SARS-CoV-2, that these physicians like Dr. Zev Zelenko and Dr. McCullough and America's Frontline Doctors and the Association of American Physicians and Surgeons and a variety of other physicians are all responding with very similar numbers. The highest credibility that a research project can achieve is not the publication of the paper. It is the demonstration by other individuals doing something fairly similar of similar outcomes and results. And that's what I want to impress upon you when I mention all these other individuals. Collectively, we have all been showing very similar results. And that similarity confirms successful treatment of SARS-CoV-2 and the successful treatment of the more progressed advanced form of what's called coronavirus disease 2019 or COVID-19 by virtue of published research, of preprints, and of multiple conversations that all of us have had together and independently as we've shared this information to make it available for you watching this YouTube video, watching other people's YouTube videos and other sites, as well as information that's been put out there on the internet for you to read. There's a lot of material that I actually looked through here in the process of reviewing this emergency use authorization. And the more I actually go through it, the more I realize that this is nothing more than redundant on what I already had covered for Pfizer and Moderna. And that's what they point out here, that despite Pfizer and Moderna having emergency use authorizations, there's no evidence that shows that the FDA has approved any product for the prevention of COVID-19. And an emergency use authorization of this vaccine doesn't advance that cause. It doesn't prevent COVID-19. It doesn't prevent infection, and it doesn't of yourself. If you've gotten vaccinated with any of these viruses, it doesn't prevent you from getting the virus, and it doesn't prevent you from spreading the virus. These are all considered, as Johnson & Johnson points out, unapproved vaccines. And for the record, vaccines are drugs. These are unauthorized, well, emergency use authorization. They're unapproved drugs. They've dropped the criteria for looking for an effect down to 30% in the confidence intervals. And a confidence interval is basically that area where you're willing to look at something and you say, I'm satisfied with this response being better than something else. 30% is kind of a low marker, particularly considering how many people are lining up for this and the proposal. They point out, Johnson & Johnson do, that there's a need for more data from Pfizer and Moderna. And for those of you who haven't gone back to the NCT website to look, the Pfizer and Moderna trials, while still active, have not been recruiting since the 20th, approximately 20th of January, one on the 20th, one a day or two before or after that. And I see nothing that suggests that they're going to be recruiting patients through the research trials. And they're certainly getting enough individuals that are getting vaccinated to see the consequences of the vaccines. Johnson & Johnson points out that there's gaps in the plans for further information from Pfizer and Moderna. Even though the committee suggested that additional studies were needed on from Pfizer and Moderna on the effectiveness in the elderly, on those that are immune compromised, effectiveness of one dose versus two doses in the schedule. And in fact, as you go through this emergency use authorization document, the same conclusions apply. There's not enough data about the elderly. There's not enough data about pregnant and breastfeeding women, lactating women. There's not enough data for children. There's not enough data for the immune compromised. There's not enough data for people with chronic obstructive pulmonary disease or people with cancer or people with heart disease. Janssen AD, the adenovirus CoV-2, is the name of the vaccine. So if you want to look it up under National Clinical Trials website, NCT, that's what you would be looking for. This is the Johnson & Johnson vaccine. It is an adenovirus vector. That means they're using a virus to infect your cells. They're taking the adenovirus, inserting double-stranded DNA made to copy the SARS-CoV-2 virus. Now you have to understand that SARS-CoV-2 is a single-stranded RNA. So what they've done is they've made the counterpart to that RNA so that they can make SARS-CoV-2 DNA, stick it into this adenovirus, and inject 50 billion of those in an injection into your arm, into your muscles, and let those go in and infect your cells, take that DNA into the nucleus of your cells so that it can make new RNA and then make the spike protein. The virus is grown in what's called PERC6 tetra cell lines. These are human embryonic retinal cell lines with bacterial transcriptional regulatory proteins. I give that to you. You can go look that up if you have any interest. This is from an originally from an 18-week-old fetus that was aborted the retina from the eye. And it's treated in such a way to provide transcriptional regulatory proteins, which is that step from DNA to RNA. 50 billion viral protein particles. They talk about CMC information. This is chemistry, manufacturing, and control, all in compliance with the FDA. This is a virus platform. So instead of using the lipid nanoparticle, they're now using another virus to infect our cells. Safety and efficacy data. Here's briefly, I want to talk about human clinical trials. There's more than one way to look at research trials, but in human clinical trials, there's phase 1, 2, 3, and 4. Phase 1 is a very small group of individuals where you really don't have any idea what dose of something you should be using or how much of it, and you test it on people starting at very low doses and going up. Phase 2 takes that dose that kind of got sorted out in phase 1 and tested in a larger number of individuals to see if it has any effect on what you're trying to treat. Now, again, the way to determine if it's effective is to determine if it helps your body's immune system make T cells reactive to the virus and B cells making antibodies to the virus. But they're not going to talk about that because that would require quantitative measurement data. And the other vaccines, Pfizer and Moderna, didn't show that. But that's the scientific method for determining actual vaccine efficacy. Phase 3 is, does it work better than what's currently out there? Well, that's an easy one to go on because none of the vaccines have been FDA approved. They're all emergency use authorization, unapproved drugs being given to people. So far, FDA has only approved remdesivir, and in our study, it only worked 28% of the time if it wasn't preceded by an aminoquinoline like hydroxychloroquine or Primaquine, which means that it underperformed chance, which would be a third of the time, just by chance that it would work. It would either work, not work, or have no effect. And phase 4 is primarily what else can we learn for it and about the drug. And that's typically the type of thing where they'll get physicians who have clinical practices. They'll pay them to continue to collect information by just prescribing the drug for people, even though it's been FDA approved. And physicians will unfortunately sometimes do that to simply have more money coming into the practice, or sometimes at the university. But universities seldom participate in phase 4 trials. Phase 4 trials are more out there in the community setting. You can see, by the way, up here that the adenovirus vector has been, they're touting it for a number of uses. They've got it out being used for Ebola, Zika, filovirus, HIV, herpes papillomavirus, malaria, and respiratory syncytial virus, RSV. Many of you know that if you have little ones who've gone to the hospital with severe respiratory distress, RSV is fairly common in kiddos, more so than older adults. You can see they've undergone a number of clinical trials here, and they go to great length in this emergency use authorization to point out the different studies that they have. The study numbers starting with 3,000 are the phase 3 trials. They've left out there's one phase 2 trial where they've looked at 660 adolescents, although the adolescents haven't been investigated yet. The phase 3 are currently still ongoing. They haven't collected all the data, but they went for that emergency use authorization. Phase 1 are represented by the 1,000 numbers. Those are all completed. Could they find a dose? Study design, they did an awful lot of information, again, focusing on the use of PCR testing, and then using that along with symptoms to determine in their estimate whether you had COVID-19 disease. Now, they really broke people down into 2 weeks and 4 weeks, 14 days and 28 days, beginning with a PCR positive test. And they had a lot of criteria for looking at it, but this is really how they defined COVID-19. Moderate COVID-19, they considered if you had a positive PCR test, and if you're breathing more than 20 times a minute, if you had an abnormal, what's called saturation, partial pressure of oxygen, SpO2, but still greater than 93%. Well, 94% and above is normal. So greater than 93 is normal. If you had chest x-ray findings or some evidence of a blood clot or shortness of breath or respiratory difficulty, or any two of the following, which they considered more minor symptoms, which you're going to have with any virus that's out there. Elevated temperature, potentially slightly increased heart rate, shaking and chills as your body's regulating, sore throat, cough, you don't feel well, you lost appetite. You've seen all these symptoms. These are the same things you have with any virus infection, and yet we haven't responded to any other virus the way that we've responded to this virus. Severe held out these criteria, which are better definitions. Actually, this is what COVID-19 is. Severe or critical corona SARS-CoV-2 infections are what produce COVID-19. So only this part of the data, as far as the definition of COVID-19 fits, really is applicable in this entire emergency use authorization. These are the people that truly do have respiratory distress. They're breathing more than 30 times a minute. Their heart rate's tachycardic. It's greater than 120, 125 beats a minute. Don't know where they get that from, but c'est la vie. An oxygen saturation of less than 94%, see, less than or equal to 93% on room air. Less than 94%, now you have a problem across the board for any disease. And this is not something you're going to know unless you're a physician. Fractured inspired O2 or PaO2. So if you're in respiratory failure, if you need to be on a ventilator or extracorporeal membrane oxygenation, where we take the blood out of you, filter it, provide oxygen to you, and then run it back into your body, that can either be venous to venous if your heart's doing well, or arterial to venous if your heart's having a problem. If you're in shock, systolic blood pressure of less than 90 or diastolic less than 60, kidney failure, liver failure, neurologic failure, you've been admitted to an intensive care unit. Well, if you're dead, that's pretty critical, and not treatable. Last time I checked. So they used a lot of primary endpoints, primarily trying to see if you developed a positive PCR test. Again, everything in this emergency use authorization that they're going for this treats and prevents COVID-19 is incorrect. COVID-19 is not the presence of SARS-CoV-2. COVID-19 is the presence of the disease producing this type of severe critical requiring treatment and hospitalization. If you had any other virus and you got put on an antiviral or you got treated for it, we wouldn't be calling you diseased. This is coronavirus disease, and that's when you get hospitalized or require significant medical intervention. So they have really incorporated people with no symptoms, asymptomatic, mild or moderate symptoms into their category to see if it has any effect. And I consider this emergency use authorization document by and large questionable because of the process of doing that. And I question the FDA in moving forward with it using this set of criteria. We have changed the target here. We've changed it from actually having coronavirus disease, and we've changed the PCR test so that we're using fewer cycles of it, so it is less likely to find the genetic material when present, which means that we will be able to better demonstrate an effect of the vaccines because you will see fewer people with a positive PCR test by virtue of the fact that you're running fewer cycles to amplify the genetic code of the swab sample taken from the nose or the mouth of an individual. These are the primary endpoints, and secondary endpoints really have to do with more disease and well, some of it's seroconversion, which means antibody titers, which means antibody titers were being run. And in fact, they admit this later on this emergency use authorization document, but again, provide no data on the antibody titers resulting from this vaccine, the true marker of efficacy. Evaluation of the safety, they go into great detail, and the monitoring of vaccine enhanced disease. Now, as you may remember, the Osaka group from Japan established its findings on the 18th of December, one day after Moderna got its emergency use authorization for its vaccine, and 8 days after Pfizer got its emergency use authorization for its vaccine, where both of those companies said there was no vaccine enhanced disease. And as you may remember from prior videos and from the FlemingMethod.com website where we talk about the virus, and we have the Osaka paper attached so you can read it. When antibodies are made to the N-terminal domain of the spike protein, it increases the ability of the spike protein to infect. That's called a vaccine enhanced or an antibody enhanced, in the case of you making the antibodies, an antibody enhanced disease. So the question is whether there's a vaccine enhanced disease. And the reality is that if you're providing a vaccine that provides for our cells to make the spike protein, and it's all of the spike protein, including the N-terminal domain, and there's nothing in this emergency use authorization that suggests otherwise, then by virtue of that, antibodies made to the spike protein will include antibodies to the N-terminal domain, which will result in antibody enhanced infectivity resulting from the vaccine, but they didn't measure that in this emergency use authorization document, or at least talk about it. They looked at the data in a number of ways, depending upon whether they wanted to look at whether they had to ask somebody if they had an adverse event versus whether they became problematic because they ended up with an infection from SARS-CoV-2 despite being vaccinated. And most of this follow-up data is 8 weeks, 2 months, which is all the FDA is asking for any of these vaccine companies to provide, is 2 months. So any statement about any longer term benefit from a vaccine cannot be made for any of the three of these. And in fact, until they show antibody titers and T cell responses, I would argue that they cannot make any statement about vaccine efficacy. And frankly, having not done the types of animal studies that needed to be done, that could have been done and should have been done, I'm hard pressed to accept evidence of human trials because it's a violation of the American Medical Association Code of Ethics, of the Nuremberg Code, of the International Covenant on Civil and Political Rights Treaty, on a variety of other laws to experiment on people before doing animal model experimentation so that you can provide informed consent to patients about the true risks and benefits of a drug, a vaccine, which is a drug, of a test, or of anything given to a patient by a physician. And I would argue that having this done at sites that roll out PCR testing or roll out vaccinations in the absence of a physician, I would argue as to the ethics of doing that when it's an experimental drug, when people are not signing informed consent. And this EUA makes no excuse or justification for that. It's limited its data to 8 weeks, even though it suggests it would like something longer. This begins to talk about the fact that individuals that are 60 and older, particularly with comorbidities, tended to have less of a benefit, with more instance of progressing on to COVID-19, and that the younger individuals tended to have more concerns about adverse events. You can go through here. There's a considerable amount of discussion. In fact, there's a huge amount of discussion about adverse events. The detail that's been put into presenting adverse events should have been, in my opinion, the detail put into presenting T cell responses and antibody titers to prove efficacy. Because as I've said before, even though we say safety and efficacy, it really should be efficacy and safety. Because if something is not effective, you would never use it. So it really wouldn't matter whether it was 100% safe or 0% safe. Nobody would receive it if it didn't work, if it wasn't effective. Some of the things that I found extremely interesting in this emergency use authorization was people getting the wrong treatment or the incorrect treatment. They were mixing up whether people were getting the placebo or the real vaccine. Now, it's not a lot. It's not a lot, but this is a research drug being given by people who know what they're doing, supposedly. So how did they end up giving the wrong thing or not knowing the right thing? This number is interesting. 10%, slightly more than 10% of the individuals who were given the vaccine turned out to test positive for actually picking up SARS-CoV-2. Clearly, the vaccine does not prevent you from getting SARS-CoV-2. More wrong dosage. Here's something, they broke down the demographics. I didn't understand this one at all because they put in the percentages and 5.4% were multiracial, so I excluded that from the numbers. There were 62.1% of the people that were white, 17.2% black, 8.3% Native American, 3.5% Asian, 0.3% Pacific Islanders, and 45.1% were Hispanic. So according to this emergency use authorization, they sampled 136.5%. That should have come up to 100% of the people, but somehow it came up to 136.5. The demographics from the countries, excuse me, that they came up with later came up to 100%. Excuse me. About 9.6% in this group that they looked out across were defined as evidence of a prior infection. So I'm not sure, again, how they define that, because evidence of a prior infection would be antibody titers. And again, they're not describing any antibody titers. So they must be collecting that data. And this would have been the efficacy data that I would have wanted to see to demonstrate that the vaccination actually did what it was supposed to do. That being said, we'll kind of scroll down through here. They did find problems with comorbidities, particularly obese individuals, those with high blood pressure or HIV infections. They were allowed to participate web-based, which might have biased some of it. If you're not web savvy or don't have computers, you're not going to participate in this type of thing. So that might have been a selection bias. Again, this is the efficacy data, and I want to point out that simply stating that somebody didn't end up in a hospital with moderate or severe symptoms is not the same thing as measuring an effective response to a vaccine. They needed to measure the T cell and B cell responses like Pfizer and Moderna. They did not report it. And again, the FDA has allowed this to slide through and simply say how many of these people ended up in the hospital sick. COVID-19 is a disease. SARS-CoV-2 is the infection. The purpose for getting the vaccination is to minimize or reduce the infection. And none of the three have been able to demonstrate that they reduce people getting infected or transmitting the infection. There's a lot of other data going through here, and I don't want to spend all day doing this. I should have checked the clock to begin with. But again, there are disparities between individuals, particularly over 75 in certain racial groups, with limited interpretability. They're displayed, but for completeness sake, they're displayed, but not some of the other data that should have been. The confidence intervals, what they're accepting as evidence, is large. It's huge. You were looking for, if something truly works, to narrow the confidence interval. And here they admit the point estimates are considerably large, particularly when there's comorbidities and smaller sample sizes from a lot of groups. We saw this with Pfizer and Moderna, where they didn't have data. Admittedly, they're stating there's insufficient data at the time to evaluate vaccine efficacy in previously infected individuals. Actually, using this particular table, it looked like the individuals that did better if they got the placebo than if they received the vaccine. Efficacy against symptomatic individuals, there were only 4 centrally confirmed mild COVID patients. So they have nothing really on asymptomatic individuals, nothing on mild. They focused primarily on moderate or severe COVID cases. And moderate ones didn't tend to be hospitalized. It was the more critically ill ones that got hospitalized, and that is mixing two sets of data here in the discussions. The severe COVID-19 cases, again, their confidence intervals, what they accepted as being acceptable for proving their point, had a wide confidence interval as opposed to narrow limitations proving efficacy. So the confidence intervals were all over the place, ranging anywhere, as you can see here, from 40 to 50 something to 90 something on a response. This is really COVID-19. COVID-19 required medical intervention. This is COVID-19 in contrast to SARS-CoV-2. And when you look at this, their confidence intervals were anywhere between -25 and almost 100%. I mean, this data, confidence intervals are huge that they're using here. This does not provide the scientific validity of a benefit to their vaccine. And these are in the people that required medical intervention. These are the ones with COVID-19, not the ones with SARS-CoV-2. SARS-CoV-2 is the virus infecting people, the vast majority of whom we know respond well, the vast majority of which we know, if they need treatment in the outpatient setting, will respond tremendously well to treatments that are dirt cheap, that have already been proven not only in the United States but around the world, and outstandingly well if they've had immunoquinolines, or if you give them to them with subsequent treatments to stop the inflammothrombotic response in the hospital. And yet we see this huge acceptable range on what they're looking at for this vaccine along with the others. Sliding through here, this again follows up that the South Africans had more of a problem, particularly if they had more than one comorbidities. They have very little information on asymptomatic individuals. So all the conversations that are out there about people that maybe have SARS-CoV-2 or the virus may even be dead, what really happens to them, and there's no evidence, no antibody data, no evidence to really provide information about what's going on with these individuals. And we know they have the antibody data because they're talking about the N-specific immunoglobulin. This is the nuclear capsid or seropositivity antibodies to non-S proteins, of which there's a variety of the other proteins, the envelope, the nuclear capsid, and a variety of other structural proteins. As specified in their statistical analysis, the secondary endpoint of efficacy against SARS-CoV-2 infection on onset of day 29 will only be tested when at least 15,000 participants with day 71 serology antibody data are available. So clearly, they didn't test enough to make the data statement about what type of immune response there is, and a secondary endpoint of efficacy against asymptomatic or undetected infection following day 29 will only be tested with all participants at least 6 months. There is a huge missing amount of gapped data not being presented. They spend a lot of time talking about the side effects and not the efficacy of this vaccine. The data shows a non-statistically significant outcome with asymptomatic. And again, they're not talking antibody titers. They're simply talking about whether people tested positive with a nasal swab. And they've got serology, but it's not evenly distributed across study sites. Wide confidence intervals, allowing them to cluster things together, lower efficacy in the newer variants and in South Africa. Continuing to slide through here, the efficacy against the variants can't be even evaluated at this point in time. Yet it's submitted emergency use authorization. But the same is true for the Pfizer and Moderna vaccines. More of the same comments, strain variants, looking at this safety, greater percentage unsolicited adverse events in individuals that are older, several reports calling for non-serious issues. Wait for the computer to catch up here. A lot of things that were omitted or incorrectly coded, particularly in the final safety and efficacy data. You can go through here if you're interested. It's easy enough to download on site. Again, these adverse events, molecularly confirmed infection, these are the PCR swabs, not included in the analysis on some of this data. Major problems were the emboli and blood clots, as we saw before. Deep venous thrombosis, pulmonary emboli, the same things that I've been talking about with the inflammothrombotic response that are killing people, was showing up in individuals who received this vaccine, the Johnson & Johnson vaccine. More on the vaccine than on placebos, and producing these numeric inconsistencies. Nice way of looking at it. Basically saying there's a significant increase in these events in the people who got vaccinated, as you can see. Some of the individuals they talked about, specifically a 25-year-old who had seizures, who had blood clots that formed multiple times, causing hemorrhage in the brain repeatedly. In this instance, a woman, a young 30-year-old, forming blood clots in her lungs, pulmonary emboli. Other people with deep venous thrombosis, and one person who developed a stroke with hemiparesis or loss of function in half their body. The data at this time is insufficient to determine if it's connected with the vaccine. Convulsions were more pronounced in individuals who received this vaccine than those who didn't. And there were hearing and balance disorders that were associated with this vaccine, and ringing in the ears, tinnitus or tinnitus, depending upon where you're at again. One particular one that had neurosensory hearing loss, completely lost his hearing. Problems with swelling, neurotic edema or angioedema, and urticaria, those are the red spots that you see on people. Problems with wheezing and arthritis and arthralgias, joints, aches and pains, and neuropathies and muscle weaknesses, all related to the vaccine, some of which had Bell's palsy that you've heard about, some of which had Guillain-Barré syndrome, which was prevalent with the swine flu vaccine. Epstein-Barr virus vaccine, instance of atrial flutter, I'm not so convinced about. It's common. Participants were excluded if they were pregnant or planned to become pregnant. Not everybody, however, got through the system. There were 7 participants who were pregnant anyway. One that got the vaccine spontaneously aborted. Other women either had incomplete abortions or elective abortions or ectopic pregnancies. The safety information is not as overwhelming as they would like it to be, and the efficacy data doesn't provide any of the antibody or T cell responses that would indicate that the body is recognizing the vaccine as promoting an immune response to provide protection against disease. Sponsors suggesting that they promote individuals who are on placebo to then go ahead and get the vaccine. So we will lose all of the placebo data, just like we're doing with Pfizer and Moderna. There's tons of missing information that they know they need to watch on. Vaccine-enhanced diseases, anaphylactic reactions or responses, blood clots, problems with missing information on pregnancy and breastfeeding women, immunocompromised patients, patients with inflammatory disorders or autoimmune disorders, chronic obstructive pulmonary disease, diabetes, neurologic diseases, heart disease, interactions with other vaccines, long-term sequelae of these vaccines, and pediatrics, less than 16 years of age. The children. They're proposing a study on pregnant women without any information on the rest of this, and they would like people to report data as to the extent possible. It's limited 2 months. That's what they're running it as. That's what the FDA has requested these vaccines, and that's what they're getting. It wasn't designed to assess any benefit in individuals with prior infections. It doesn't prove that you don't get infected. It doesn't prove you can't transmit the infection. the limited access and information about asymptomatic individuals, no long-term sequelae. One of the things that kept coming up was myocarditis or inflammation to the heart from the virus. Um, there's not even the type of data they need for mortality, although they know they need to get more data on it. Vaccine effectiveness, raising questions about even after the vaccine, the need for persistent mask wearing and social distancing, um, and questions about viral shedding and transmission to individuals. All of this raises lots of questions, again, about the same types of things we've seen before. Um, the vaccine enhanced disease, again, now this actual vaccine company has no right to pretend that it submitted its EUA a day or 8 days before the Osaka paper, because the Osaka paper is out there. You can find it. You should be familiar with it. If you're doing research or submitting a drug, either a vaccine or some other treatment for SARS-CoV-2, you should be familiar with the literature to actually submit. So with that, I'm going to stop screen sharing and simply point out that the information in the emergency use authorization for the Johnson & Johnson vaccine, which is a nanovirus double-stranded DNA designed to insert into the nucleus of your cells to transcribe then messenger RNA to then translate and make spike protein, with all of the problems that we've talked about with spike protein in the past, being released in the bloodstream, crossing the blood-brain barrier, causing the neurologic effects, causing potential prion-like disease abnormalities, including potential for amyloidosis with heart disease. All of that still remains as a contributing concern. There's no evidence that T cell responses or antibody titers provided in this. They have confused or conflated, put together the concepts of the infection with SARS-CoV-2 with the actual coronavirus disease itself, and tried to mesh that data together, all the while still missing large pieces of information. But yet, submitting an emergency use authorization that, based upon what I saw, is going to fly through the FDA, because there's a demand for vaccines under the belief that this will somehow change this endemic. Not a pandemic, it's endemic. Now, the things that are responsible for changing an endemic or even a pandemic are adequate treatments. It is disheartening that the amount of money that has been spent on vaccines was not invested into research to find treatments that myself and numerous other individuals have been able to do and provide evidence on in publications, sometimes preprint for some individuals, but in discussion, very credible data, and data backed up by consistency from one study to another. Hopefully that provides some insight. I think that was kind of a rapid addressing of the emergency use authorization document that was submitted yesterday by Johnson & Johnson to the FDA. It will undoubtedly receive approval in record time, if it hasn't already by the time I finish this recording, but I wanted to present it to you so that you would have an opportunity to see just some of the information that I was looking at, as well as my concerns with this particular vaccine. Again, emergency use authorization when treatments exist isn't a recognized concept. The utilization of vaccines that are still experimental are not what I consider an accepted scientific approach. The absence of animal data and the investigational approval of this emergency use authorization increases the number of people that they will have experimental data on, but it still makes it an experimental drug being used on Americans and people around the world, along with the Pfizer and Moderna vaccines. I'm not anti-vaccine. If we could find a vaccine that would actually have the desired immunologic effect without adverse effects, that would be scientifically outstanding. From a scientific and medical standpoint, I'm not sure why we need that when we have already demonstrated successful treatments. This is Dr. Richard Fleming. Today is Saturday, the 27th of February, 2021.